Alright, everybody. Hello, and welcome to Lecture 7. This is an exam week for you guys. I do want to make an announcement that I will be recording a video for Lecture 8, but I understand that you guys that are taking your exams in, spring 2026 do not have exam material from Lecture 8, so I wouldn't be too concerned about that. I'll reiterate that when I actually do a video on it, but that one's not going to be useful for those of you who are taking this class in the spring of 2026. Okay, so let's go ahead and take a look at this stuff. So I'm not going to go over the questions from past quizzes and stuff. I don't really have time. And yeah, so there's a lot of it, to be fair. I do think this slide's pretty important, so just so you know, very briefly, regulated is when an external signal or something kind of assists in the pathway, and then consecutive is just when it kind of happens randomly, almost, if you will. That's kind of the main thing I would take from that. Okay, so now we're going to talk about cell signaling. So cell signaling GPCRs is the main topic here for today. You guys have already seen what cell signaling basically looks like. You have a cell, you have the cell releasing some sort of signaling molecule that binds to a receptor on a different cell, and then that cell does some weird stuff. And so here's another example of that. I wonder if they show, yeah, so here you can see that, oh, okay, this receptor binds to this ligand, it leads to a bunch of signal cascade things, and these things lead to more signaling that cause transcription or cause all sorts of other random stuff. And then these can mess around with many different things. They can change metabolism, expression, shape of the cell, a whole bunch of stuff. I would remember these types of signaling. They are not hard to memorize, so I would not recommend writing them on your note card. All you have to do really is contact dependent is obviously when they're in contact with each other. So maybe there is a cell signaling molecule stuck on one cell, and it is able to physically bind to a receptor on a different cell. Paracrine signaling is when you have a cell releasing some sort of molecule and that binds to a receptor. That's paracrine. Synaptic is like a subcategory of paracrine. It's just that synapses are a little bit closer to each other, so it's kind of differentiated just a bit due to that. And then endocrine is honestly my favorite one. Endocrine is when signaling molecules are released into the blood. They then travel through the bloodstream and then they get dropped off at a cell. So this endocrine is for very long distance travel, while contact dependent is super short distance, and then paracrine and synaptic are maybe medium distance, if you will. So, there's a couple different types of receptors. You guys have probably seen these at this point, but you can have cell surface receptors, which are on the membrane, or you can have intracellular receptors, which are within the cell and often within the nucleus. So, what makes these different is you need certain types of signaling molecules to do either. So, for cell surface receptors, you'd probably be binding peptides, so protein chains or amino acid chains, because those are polar, right? They don't really, aren't able to pass through the membrane. But for an intracellular receptor, you would bind something like a steroid. So testosterone, for example, or estrogen, those are steroidal hormones. And so those are nonpolar and therefore have an easy time passing through these membranes to bind to these receptors. So you can think of it like that. So think of so insulin, for example, is a cell surface receptor. It is a peptide. And then testosterone and aldosterone and estrogen, those are all examples of steroid hormones. They use intracellular receptors, generally speaking. So, this is basically just a very chill introduction to what spatial summation and temporal summation is. It's obviously not what that is, but it's a very similar concept. Combinatorial signaling is the idea that signals from two different cells can interact with each other. So if you have one cell and it's being fed different signals from two different cells, both those signals will combine to form what the what the main cell is able to see or hear, if you will. Don't really like personifying it, but I can't think of a better way of explaining that. So let's say we have an antagonistic signal. Signal 1 and signal 2 both come in at relatively similar time points. If it's just signal 1 alone, signal 1 has a large response. But if you add signal 2 for it, it goes down. This suggests that whatever signal 1 is doing, signal 2 is counteracting it. That's all there is to it. For synergistic ones, it's the opposite. So signal 1 alone is small, but when you add them together, they become quite large. So that right there is an example of synergistic. It's like this doubles basically, while this halves, if you will. It's actually less than half, but whatever. Here are some examples of cell signaling. You do not need to memorize these, I would like to think. So, insulin, I talked about insulin, peptides, a protein. So you should be able to think, where would these all bind? Where would they bind? So insulin, for example, would probably bind to a cell surface receptor because it is amino acids. Thyroid hormone, this is a bit of a tough one, but a lot of polar stuff going on here. So perhaps it would bind to a receptor, right? It seems pretty polar to me. Thyroid stimulating hormone, amino acids, so it's going to bind to a cell surface receptor, probably. Thyroid releasing hormone, 3 amino acid peptide, so again, a cell surface receptor. Adrenaline, pretty dang polar, I would say, so a cell surface receptor. Histamine, histamine's a bit of a weird one. Probably would say it's going to be a cell surface receptor. It seems kind of polar, but I could argue both ways maybe for that. Nitric oxide is pretty small, kind of polar. So this one's also kind of tough to tell. So maybe this would bind to a cell surface receptor, but because it's super small, I could see the argument for maybe it binding to, what's it called? To a cytoplasmic or nucleic receptor. Although I think this actually should have a charge on it, no? I think there should be a charge on that. If there's a charge on it, then it shouldn't be. Oh no, never mind. It's a radical. So yeah, it's a bit of a weird one. This is a bit of a strange one. I think this is actually a cell surface receptor one. I can't remember off the top of my head, but you basically should be able to kind of infer what the type of cell signaling would be based off the structure, if you will. That's what I'm trying to get at. So let's look at endocrine signaling. Thyroid. You guys have already seen this in BIOC 220, hopefully. And if you haven't, you've probably seen it if you studied for the MCAT. If you're taking the MCAT already, you've definitely seen this. Very important pathway for metabolic rate. So thyroid signaling, the main thing here is the hypothalamus sends a signal to the pituitary in the form of TRH, so thyroid releasing hormone. Thyroid releasing hormone binds to the pituitary, specifically the anterior pituitary, I believe, which then releases thyroid stimulating hormone. And this thyroid stimulating hormone goes through the blood and binds to cells in your thyroid. And your thyroid releases thyroid hormones, which are T3 and T4. And T3 and T4 bind to target cells and increase their metabolism. Here is a example of a disease. So again, the normal pathway is hypothalamus TRH to pituitary to TSH to blood vessels to thyroid cells. Those thyroid cells make T3, T4 thyroid hormones, and those thyroid hormones bind to target cells, which increase metabolism. And so here's an example of Graves' disease. So Graves' disease is actually a disease that binds. Where is it? Yeah, okay, yeah. So Graves' disease is basically there's an autoimmune response where the antibody hyperstimulates the TSH receptor. So the receptor on the thyroid cells, on these cells, is hyperstimulated. And so what ends up happening from that is this antibody binds to the receptor. It's always active and so the thyroid cell releases a bunch of T3, T4, and that it's just too much into the bloodstream, and so your cells pick up this T3, T4 and become extremely hyperactive in terms of metabolic rate. So you like burn through food, you burn through energy ridiculously fast. For paracrine, there are 3 classes of surface cell receptor, and you should know these. There are ion channels, which you've hopefully seen from BIOC 220. Basically, a ligand binds, they open, and then ions flow through. There's G protein coupled receptors, where you bind a ligand. This activates this G protein complex by switching out a GDP for a GTP. And when that switches out, the G protein shifts, and this active part of it is able to do some other functions like activate adenylate cyclase and other signaling cascade molecules, or signal cascade proteins, if you will. Enzyme coupled receptors are receptors that have enzymatic portions on them. So there's like 2 monomers right here, basically. And then what you do is you bind the signal molecule, which is usually a dimer of some sort, and the binding of the signal molecule causes these two to dimerize together, causes these two to come together and dimerize. And these are normally inactive catalytic domains, but when they combine in the dimeric form, they form an active subunit. Another example is, maybe you have 2 pieces that need to come together for a separate enzyme to actually bind to this receptor and become active. So these 2 are inactive, they're not working. The signal molecule binds to them to form this actual receptor, and then this is able to house a specific spot for an enzyme. So something like that. Okay, G protein coupled receptors. This is what a G protein coupled receptor looks like. To very briefly, G protein coupled receptors are 1, 2, 3, 4, 5, 6, 7 transmembrane domain proteins. So, if you guys remember from the previous lectures where we talked about how things are put into the ER and like the orientation of them to get them in a certain way, this thing has 7 different transmembrane passes, so it definitely is going to have like 7 different signals. That's something you should be thinking about, actually. And also, is it going to have a start signal sequence? Is it going to have where is it going to have that start signal sequence? Does it matter? Maybe it differs from protein to protein. Okay, the structure of an inactive G protein. So usually the actual receptor itself is on this side of the membrane. So the receptor would be like on the extracellular matrix, so out here. And the inside is this actual G protein. So this is the G protein. The G protein has 3 different domains, an alpha, a beta, and a gamma domain. And the main thing to keep in mind for these G proteins is the alpha domain, aka the RAS domain, is the portion where the GDP and GTPs bind. The beta and alpha subunits, beta and gamma subunits are usually the ones that do some sort of signaling, but that's not always the case. So here we have an inactive G protein, right? So we have an inactive GPCR and we have an inactive G protein. What's going to happen is first, we're going to bind a signaling molecule to this GPCR. That will cause this inactive G protein to bind to the GPCR, because the GPCR has a conformational change. So you look at this portion here. See how it becomes circular a little bit? So this is inactive, ligand binds, causes a conformational change in the receptor. The receptor is able to bind the entirety of the inactive G protein. When that happens, the G protein itself undergoes a conformational change and releases this GDP from this AH domain. And this is a nice little thing here. The GPCR serves as the Jeff. So basically the GPCR itself is not the one actually doing the exchanging, but it basically acts like one, if you will. It does stimulate this process of replacement. So here you see GDP leaves, and what comes in? A GTP. And so when this GTP comes in, everything kind of splits from the receptor. So the GTP comes in, this splits off, this also splits off, and these can do different signaling things. So I know I said usually that these are the ones that do the signaling, but sometimes the alpha and the beta gamma do separate signaling. So it's very possible that you could have many of those cases. So see here how these all split. So now you have that. And so here's the thing, a regulator of G protein signaling will serve as a gap. So basically it'll hydrolyze as GTP, and when this thing hydrolyzes to GDP, all of these kind of come back and form together, and then a separate process is needed for the actual receptor with arrestin and something else. But oh yeah, here we go, perfect. So yeah, what needs to happen to turn the pathway back off? So we talked about a lot of these, right? You've got to hydrolyze the GTP. Then using the RGS, right? So the RGS is like a separate protein usually that helps, that binds. You have to inactivate the alpha, which usually happens when you hydrolyze the GTP. You also have to dissociate from target protein. I'm not too sure what that's trying to get at there. Yeah, I'm not too sure what this is trying to get at here exactly, but this is basically in order. Hydrolyze GTP using RGS. Alpha is inactivated. Inactive alpha reassembles with beta and gamma. And the inactivation of the receptor by ligand release or desensitization. Or intake into the cell, actually. I don't actually know what they want here. Dissociation from target protein. It's a bit of an interesting way of phrasing that. Here we go. Desensitization. Arrestin. So this is a very interesting part of GPCRs. A lot of GPCRs actually bind so strongly to their ligands that it's impossible for them to be removed. Or they don't get removed very easily. So the cell has come up with a separate mechanism to remove them, which is cell-mediated intake / uptake, whatever you want to call it. So the way this works is when you have an activated GPCR, even after, even if like the G protein units have maybe become inactive, a lot of the times the GPCR portion remains active, which is a kind of a problem. You don't want it to be active the entire time. So what ends up happening is with this ligand, the ligand bounds, leads to this conformational change, which we saw usually binds a G protein. However, it is possible that instead of binding a G protein, it binds I don't like how this is named. Yeah. Anyways, it stimulates the process of a GPCR kinase, GRK, to actually phosphorylate it. So if this is exposed enough, you can imagine that, okay, too much exposure, it'll get phosphorylated eventually. So eventually, it'll get phosphorylated by GPCR kinase. Gets phosphorylated 3 different times, so you're going to need like 3 different ATPs for this. And then once the 3 phosphates bind, that is the cue or the recognition for the protein arrestin to actually bind. So arrestin will bind to this triple phosphorylation site, and it'll cause the endocytosis of the receptor. Yup. This. Basically, if you use opioids too much, a lot of the opioid receptors will actually be endocytosed by the cells to try and mitigate the response, which is why people end up taking more and more of them to feel the same high, if you will. GPCRs, arrestin also facilitates receptor recycling. So, again, we talked about this already. So, here's a case where you see normal GPCR, this thing binds arrestin, then this gets coded. These get coded, right? coated, and then it gets endocytosed. Once it's endocytosed, it's put into an endosome, and maybe, you know, what's necessary for the ligand to dissociate from the GPCR is like a low pH or something. You never know. So the endosome does some magic with enzymes and pH maybe, and causes dissociation. And then these receptors can actually be recycled. So these can be recycled and put back on the cell surface. And the ligands can actually be degraded and used for other cellular processes. And so yeah, usually when the receptor is released, or sorry, when the ligand is released, the arrestins also get released. So you can see here in this step right here, this step here, we go from having arrestin and ligand to just the protein. So the arrestin and the ligand basically come off at similar times in this endosomal sequence. Okay, cyclic AMP. Cyclic AMP is a very versatile second messenger. Does a lot of things, regulates TSH secretion, adrenaline, glucagon. Basically, all these peptide hormones tend to have a pathway with cyclic AMP involved somehow. And the way that happens is through a thing called adenylyl cyclase, or adenylyl cyclase. And so basically what happens with most of these adenylyl cyclases is when you activate a GPCR, the alpha subunit with its GTP actually bind to the adenylyl cyclase and cause this conversion of ATP to cyclic AMP. And in case you're curious of what the process to make cyclic AMP looks like, it's basically just chop off these two and this nucleophilic attacks here. I'm going to say it right now, you are not going to be asked draw the process of forming cyclic AMP. That is too basic, and it really isn't super high yield, I would say. So I wouldn't be like too concerned about how it happens, just more so that it does happen. Do keep in mind a little bit of how the mechanism functions, though. I think it is helpful to know that there's a nucleophilic attack in here, just trying to remember some OChem knowledge, So here's an example, actually, of cyclic AMP being a second messenger. G protein activates adenylyl cyclase, form a bunch of cyclic AMP. These things bind to a thing. So usually there's a pKa. So pKa is protein kinase A. This is usually bound to these inhibitory subunits, so regulatory subunits, these things. These things have binding sites for cyclic AMP, so what happens is these bind to these regulatory subunits, and that causes conformational changes, which causes these things to split from each other. Once they have split, the active site is revealed. So the active site is like kind of like maybe like this, right? It's like hidden inside the regulatory subunit. You can think of it like that. So once these split, these are now open, and so these are now available to be used as what's it called? I can't think. Those are now available to be used as correctly, or as activated enzymes, not correctly. So yeah. So yeah, here we go. Here's another process of this whole thing. So back to inactivation of it. So inactivation, we talked about a couple of these. So these three we talked about. So the signal needs to be internalized and degraded. So if we want to stop this process from happening, we need to delete this receptor, basically. We also need to hydrolyze this GTP. So this GTP needs to become GDP. And yep. Now, the other thing that we need to keep in mind is these secondary messengers. We also need to be able to get rid of the secondary messengers quickly so they don't cause downstream effects. So the way that we do that is there's an enzyme called phosphodiesterase. What that does is it breaks down cyclic AMP into just normal AMP. So that stops that. And eventually what happens is all of this cyclic AMPs will dissociate from these regulatory subunits, and these catalytic subunits will be able to rebind the regulatory subunits. And then after that, there are also going to be phosphatases downstream of the pKas that inactivate the targets that pKa phosphorylated. Because remember, pKa is protein kinase A. It's a kinase. You probably remember seeing this from like pyruvate dehydrogenase. It's actually a regulatory enzyme for PDH. But yeah, so activated pKa, right? So the activated pKa is going to phosphorylate things, but there's going to be phosphatases downstream that remove that phosphate and deactivate the things. So, you guys have seen this like 8 million times. Glucagon you guys are very familiar with. Adrenaline actually uses pretty much the exact same receptor pathway as glucagon. It's a bit different if I remember correctly, but it's pretty much the same. You guys know that if you have too much glucose, you use insulin to remove glucose from the blood. If it's too low, you put glucagon in there, and it breaks down glycogen, puts glucose into the blood. Adrenaline is very similar, but it's released from the adrenal glands, but it has a very similar function. The adrenal gland is actually probably one of my favorite glands, I'm not gonna lie, or favorite endocrine organs. Physiological response to adrenaline. Okay, so here we're gonna look at what this looks like. So we have an inactive GPCR, inactive G protein, inactive adenylate cyclase. We then bind a ligand. In this case, it's going to be adrenaline, which is going to cause the G protein to become active. This G protein is then going to split. The alpha subunit is going to be the one doing the signaling in this case, and the beta and gamma kind of just chill. The alpha subunit binds to the cyclase, causes cyclic AMP production. Cyclic AMP binds to protein kinase A, binds to the inactive pKa regulatory subunits, causes release of the subunits, resulting in active pKas. These active pKas and then start binding to a bunch of pathways. So this active pKa first phosphorylates glycogen synthase, which inhibits it, so you cannot make glycogen. It then binds to it then phosphorylates phosphorylase kinase, which makes it into an active phosphorylase kinase. And then phosphorylase kinase phosphorylates glycogen phosphorylase, which if you remember, glycogen phosphorylase is actually the enzyme that causes glycogen breakdown, so the first step of that. So you then activate glycogen phosphorylase, which turns glycogen into individual glucose 1-phosphate monomers. So, twofold response. It inhibits glycogen synthase and activates glycogen phosphorylase. That's the main thing you need to keep in mind. You do need to memorize this whole pathway, though. You should know the entire thing. Put on your note card if you must. I think the difficult part is kind of like, people often forget this portion with the phosphorylase kinase. So just remember that, and you should be good to go. So for the inactivation, you just have to reverse all these, right? So you have to remove the phosphatase, remove the phosphate, remove the phosphate, remove the cyclic AMPs from these, bind all these together again, remove the phosphate from this, hydrolyze this GTP to a GDP, combine all of these together, remove the adrenaline through some sort of endocytotic pathway or something like that. So there's a bunch of things that you can do, right? I'm not going to go over every single one. They're all listed here for you if you want to look at those, but you can kind of just work backwards. So you can actually see, like, I talk about everything here. So internalized, hydrolyzed. I guess I didn't talk about this. This is an important one, actually. Cyclic AMP needs to be turned into AMP by phosphodiesterase, so that's an important one. Talked about this. Sure, you can already see that. That's how everything ends up anyways, so. Okay. pKa, also has other functions. It doesn't just do this. It can do other things as well. So another thing that it can do is once PKA is activated, PKA can actually go into the nucleus. It can diffuse into the nucleus, and it can activate a thing called CREB. CREB is, I can't remember what it actually stands for. Tells you right there. Cyclic AMP response element binding protein. It's not because cyclic AMP itself binds to it. It's just that it's part of a cyclic AMP pathway where cyclic AMP activates PKA, or cyclic AMP binds to a regulatory subunit, and that causes the removal of it from PKA, which makes it active. And then the PKA phosphorylates CREB, and then CREB, once phosphorylated, will bind to this thing called the CREB binding protein. And then once the CREB binding protein plus CREB are like attached to each other, they bind to these different areas of the DNA called cyclic AMP response elements, which upregulate or downregulate different genes. Yeah. So this is just a pathway to show that, you know, these signals can also be taken into the nucleus and affect transcription as well. They don't have to just affect cytoplasmic processes. Also, something you should remember this. This is a very important pathway too. Please remember all this. All this is trying to say is that CREB-dependent genes are transcribed in this environment, but that it also includes things like survival genes. So BCL2 is actually it's a cell division gene and cell growth gene, if I remember correctly, and then other metabolic genes. So all sorts of things can happen. Ion pumps. I think it's actually very important that you do remember these concentrations, or at least try to write them down somewhere. The main thing you should know is that concentration gradients always go from high to low, and we don't really care about what's it called in this class, the electrical potential of the membrane right now, so I'm not going to worry about that too much. But you should definitely try and keep in mind the concentrations. So, you know, extracellular for sodium and intracellular, all that jazz. So, yeah, but ion pumps also regulated by cyclic AMP. So ion pumps, remember, are things that push things against their gradient. So sodium, for example, sodium channel would go down its gradient, and channels, if sodium's high on the outside, where is sodium? High on the outside, it's going to want to go into the cell. So it's going to go into the cell. Potassium's going to go from 140 to this, that way. And then chloride is also going this way. So you can see here, a channel goes with its gradient, a pump goes against its gradient. So if it's high sodium on the outside, low on the inside, a channel goes down the gradient. For the pump, if it's high on the outside, low on the inside, it's actually going to go against the gradient. And this usually uses some form of energy, maybe ATP, maybe some other form of energy. There are many different types of types of pumps. This is one of them. So there's a calcium ATPase. This pumps calcium back out of the cell, if you will. So, and it uses ATP. There's a sodium potassium pump. This pumps sodium out of the cell and potassium into the cell. This also costs ATP. And this is actually a fun one. The antiporter is not an ATP requiring pump. What really is interesting about it is that sodium goes down its gradient, and the energy from that is used to push calcium against its gradient. So sodium is going down its gradient, that releases energy, and that is enough energy to push one calcium against its gradient. So, cool thing about this, olfaction, so your sense of smell, and taste actually to an extent, depends on cyclic AMP regulated sodium pumps. So what happens is an odorant molecule, which is just an odorant is just a chemical that comes from a food, basically, binds to the GPCR, which activates it. Again, we switch out a GDP for a GTP. It's not shown here. A GDP for a GTP in the G protein. The alpha subunit binds to adenylyl cyclase. The adenylyl cyclase changes ATPs to a bunch of cyclic AMPs. And the cyclic AMPs bind to these ion channels, these ligand-gated ion channels, which lead to sodium influx into the cell. And that sodium influx leads to not an action potential, but it leads to a bunch of graded potentials, actually. I actually want to double check something. Is it action potential? I can't remember. Oh, my bad. I lied. These do have action potentials. Sorry. So the sodium binds here. I think I'm thinking of rhodopsin, so like rod cells and cone cells. Those do not have action potentials. Sodium, though. So sodium comes in, leads to an action potential, and that results in neurotransmitters being sent to the brain. So this is another pathway I would try and memorize. But that's actually going to be the end of this video. I do hope it was helpful for you guys. Not too long, actually. So, we take those. But other than that, good luck on your exam, guys, and happy studying.